Hello everyone, this is Melba Academy and today we will be solving a problem from the Hardy-Weinberg equilibrium. So this problem is composed of cats. So reading the question, we determine that there are two colors of cats, the black cats and white cats. So the black cat allele is represented by capital B, which is the dominant allele. So according to the equation of Hardy-Weinberg, it is presented by P. And the white cats with allele B, small b, are the recessive alleles, and they are presented by small q. So we have a population of 1000 cats, which consists of 840 black cats, 160 white cats. And we have to determine the allele frequency, the frequency of individual per genotype, and the number of individuals per genotype. So, as we know that there are three different genotypes for such questions, according to Hardy-Weinberg, we have the homozygous dominant, we have the heterozygous dominant, and we have the homozygous recessive. So we will be calculating the genotype frequency of the homozygous recessive. Why, you may ask? Because there is only one genotype for the recessive allele. The frequency of these both individually cannot be determined, as we have a heterozygous allele and a heterozygous individual and a homozygous individual. But here we only have one type. So first we will be determining the frequency of individual per genotype. The genotype is small b and small b. So the frequency is found by number of individuals divided by the total number of individuals in the population. And as we read the question, there are 160 white cats, which are the homozygous recessive individuals. So we will write 160 divided by the total number of cats, which is 1000, and our answer will be 0.16. And as we learned from the Hardy-Weinberg equation, for genotype, the genotype is represented by the genotype of homozygous recessive individuals is represented by q square. So we know what q square is, it's 0.16. So with the help of the genotype of homozygous recessive individuals, we can find the allele frequency. So allele frequency is q without the whole square. All we have to do is remove the whole square just by taking the square root. And our answer will be q equals 0.4. So we have q square, we have q, and with the help of the equation, we are now going to find p. So we know the equation, which is p plus q equals 1. So we know q and we know 1, obviously. So we'd find p with the help of this. P is equal to 1 minus q. Q, as you can see, is 0.4. And our p is going to be 0.6. So we have found q, we have found q square, and we have found p. And p is the allele frequency of, it's the allele frequency of the dominant allele, which is capital B, whereas q is the frequency of the recessive allele, small b. And q square, it is the genotype frequency. of recessive individuals. So we've got all of this, and we just have to find q square. P square. That's pretty easy to find. We'll just take the whole square of this, and p 0.6 square. P square is going to be 0.36. Now we have found the genotype frequency of the dominant allele, the dominant individual. We've got all four of these. All we are left with is the heterozygous individual. As we all learned from the equation, we know that p square plus 2pq plus q square is equal to 1. So we know that the heterozygous frequency can be found by multiplying the frequency of dominant allele with the frequency of recessive allele. So as we know that 2pq is, the p would be 0.6 and q would be 0.4, so we get 0.48. So what did the question ask? The question asked for allelic frequencies I'm going to write them all again just in case anyone's confused. So allelic frequency, we have P and we have Q. P was 0.6 and Q was 0.4. And the genotypic frequency is P square was 0.36, Q square was 0.16, and 2pq is 0.48. So what's left? The last portion of the question asked us the number of individuals per genotype. So we have to tell them how many individuals were homozygous dominant, how many individuals were heterozygous dominant, and how many individuals were homozygous recessive. So we know that homozygous recessive individuals were 160 white cats, but we don't know how many, like we're not sure how many cats were heterozygous dominant and homozygous dominant. It is quite easy, if you ask me. All you have to do is take the genotype that you want to find out and multiply it with the total population, and you would get 0.36 into 1000 cats. We get 360 black cats which were having the homozygous dominant. So that's how you solve a Hardy-Weinberg problem. If you have any constructive criticism or any other questions that you'd like to ask, please let me know, and make sure you like and subscribe this video and share it with your friends. Let them know how to find a frequency. But yeah, so thank you for watching.